Good morning, everyone, and many thanks for joining us for this webinar today. I'm Donald Murphy. I'm an archaeologist with ACSU, and I'm your host this morning. This event has been carried out as part of our CPD programme for the N55 Corduff to south of Killydoon Road scheme in association with Cavan County Council and TII. I'm joined this morning by my colleagues, Dr. Kerry Cleary, who organised the event, Mr. Niall Roycroft, Project Archaeologist with TII for the scheme. I'm particularly delighted this morning to welcome Dr. Jessica Smyth, Assistant Professor in the UCD School of Archaeology, whose previous research has included organic residue analysis of Irish Neolithic pottery and the Neomilk project that explored the introduction and spread of cattle-based agriculture by the earliest farming groups in Central Europe. Dr. Smyth is currently the principal investigator on the Passage Tomb People project, investigating the social drivers of Passage Tomb construction in the 4th and 3rd millennia BC. Today, Dr. Smyth will be speaking about some scientific techniques that can potentially be applied during our post-excavation analysis, including isotopic analysis of bone and collagen, molecular fingerprinting of collagen peptides, and lipid residue analysis and dating of lipids. The format for today will be that Dr. Smyth will give our presentation first, and we will then have time for a short question and answer session afterwards. To keep some order on proceedings, all attendees have been muted for the minute, but you should all have access to a question and answers button at the bottom of your screen. If you have any questions as we go along, please feel free to type them in there and Kerry will collate them and we'll ask them during the question and answer session at the end. If we still have some time at the very end and there's something that you feel hasn't been addressed, you can raise your hand using the button at the bottom of your screen and I will bring you in and you can ask your question. Just a quick note that this presentation is also being recorded for future dissemination. So without any further ado, I'll hand you over to Dr. Jessica Smyth.
That's great. Thanks a million, Donald. Delighted to be talking this morning. I'm just going to share my screen now. I'm going to my presentation. There we go. Okay. So there we go. Yep. So morning, everyone. As Don said, his introduction, I'm going to talk to you a bit this morning about archaeological science methods and how we can apply them to excavated material that you kind of typically find on a range of Irish archaeological sites. I thought the most useful way to do this was to do it through the frame or the lens of the project I'm currently leading, the Passage Tomb People project, just to show you kind of in practice how these different techniques can mesh quite nicely together and the potential of them. So basically, as I said, I'm going to be giving you kind of a bit of an overview this morning on the various biomolecular and biogeochemical methods available that you can use on excavation material through this project, which was or is an Irish Research Council laureate grant awarded in 2018, and that runs till kind of autumn next year. So we're nearly 3/4 of the way through. So I'll just start by giving you a brief background to kind of the project setup, the kind of reasons behind it, and some of the questions that we're tackling here, which are similar to questions you might want to be looking at with your kind of individual sites on schemes such as the N55. I'll give you a brief intro into the methodology and why kind of molecular level analysis is particularly good for Irish archaeological remains and kind of the state of preservation we generally find them in. And then I'll finish with two kind of works in progress, so two kind of aspects of the project that we're getting really nice results from at the moment. So yeah, just a bit of a background to Passage Tomb People. I suppose the rationale for writing this project and applying with this project was that even though monuments such as kind of Newgrange in Louth and other monuments in the West are such iconic monuments and so kind of there in the public consciousness. When you kind of dig down into it a bit, we very little really idea of who these communities of builders were and what worlds were they part of, and kind of crucially for me, what types of resources allowed these amazing feats of engineering to take place in the first place. So what sort of subsistence was kind of underpinning these ways of life, that they could kind of take time out to knock some of these things up. I'm sure loads of you are familiar to the broad timeline, but just in case you aren't, we're kind of talking about this phase of activity that is several centuries after the arrival of farming in Ireland. So something is happening in those hundreds of years between kind of these resources being imported, such as kind of domesticated cereals and animals, and this almost ramping up to, at the end of the fourth millennium, very, very elaborate megalithic architecture. So I wanted to know, or was curious about finding out more about kind of what's underpinning that. So here is the project, and here's some of the project team, or all of the project team. As I said, this is the as a research scientist, and then we're collaborating with team members in Bristol. So we've got PhD student Lily. I'll talk a bit more about her work at the end of the talk, with her supervisor, Professor Richard Evershed. We have Richard Madrick in Cardiff, who's overseeing the light isotope analysis, Marie Bellas in Paris, who has pioneered sequential sampling of animal teeth and their modelling, and again I'll talk about that in a few minutes. We've Mike Buckley heading up the proteins analysis in Manchester, and we've got prehistorians and zoo archaeologists also based in Orkney College, which are helping us with the Scottish material. So so this is the project study area. Passage fumes, again, I'm sure you're very well aware, are kind of, broadly speaking, an Atlantic facade phenomenon. So going all the way kind of from the tip of Scotland all the way down to southern Spain. But this project is focusing on kind of 3 target zones across the Irish Sea zone, or Irish Sea area. So Orkney Islands, Ireland, and Northwest Wales, primarily focused in the area of Anglesey. And I picked those 3 areas because we know culturally they're very closely connected, kind of temporally very closely connected, and it allows us to kind of zoom in on the mechanisms between those communities and how they were influencing each other. Were they doing similar things to enable them to build the monuments that are appearing in their areas? As I said in the intro, kind of crucial to this, or kind of at the core of it, is already excavation material. So we're not digging up any new sites, we're not undertaking any kind of field work in that sense. We are going to museums, archives, institutions, and working with the stuff that's already out of the ground. So here's kind of the relevance from material that might be uncovered along the N55. So a bit about research questions, and I think when you're undertaking this sort of kind of wide analysis or kind of large scale analysis, it's really helpful to kind of frame your research questions first, and what do you hope to achieve or to contribute or to understand better by doing all of this sorts of stuff. Because you can have huge budgets for scientific analysis, you can throw kind of the kitchen sink at it, but if you don't know what you expect to get out of it, it's kind of a waste of money. So the research questions I primarily focused around: did farming fail after the early Neolithic? You'll see here, these are just some screen grabs of recent enough papers talking about there were the early farming kind of in the early 4th millennium in Britain and Ireland, and some evidence or some suggestions that kind of the population crashed, or there's a kind of reduction in evidence on the ground of people doing stuff kind of in the centuries after 4000 BC. You'll see here, these are just radiocarbon dates which have been summed and modelled, and the authors here are suggesting, well, the blue arrows here are where kind of farming arrived, so around 4000 BC, and this kind of slope is the kind of modelled population curve, or what a population would look like as it kind of increases naturally. And they're saying there's loads of radiocarbon dates kind of right after farming. So they're saying it's a population boom. Farming is brilliant and everything goes well, but then they identify in the radiocarbon dates this kind of trough, and they've interpreted that as a population collapse or a bust, so kind of a failure of the farming experiment. And that kind of rang a bit weird for me, because I know obviously this is the period where you have these big passage tombs starting to be constructed, but also, and this will be a familiar sight to lots of you, no, kind of based on my earlier research, my PhD research, while I know these early Neolithic settlement remains are really visible in the ground and they're very well recorded, they're very well dated, what comes after them is quite sketchy looking, kind of ephemeral looking, archaeologically speaking, which sometimes doesn't get those same amount of resources and definitely doesn't get the level of radiocarbon dates, for example, that you might put into a lovely rectilinear timber house. So kind of something about that was a bit fishy for me, and I wanted to explore that in a bit more detail. So it's basically this sort of scenario where we have the early Neolithic being really visible on the ground, either through settlement remains or through things like evidence for cereals, evidence for animal bone, lots of radiocarbon dates. And it was really about investigating this apparent lull or trough or difference. What's going on here? What is sustaining these communities that are building passage tombs? And yeah, that, like, in terms of the challenges, well, you're probably all aware of them. One big challenge is scale. Passage tombs in general, well, especially the kind of the super big one, and this is kind of Nouth over on the right here, they're very large. The excavation of which is a very big and long undertaking. So, you know, Nouth is still being kind of published half a century after it was excavated. And what tends to happen with these very big projects is that the analysis is, you know, it's quite site specific. So it's focused on fully understanding maybe a single site, and the linking up of the different sites, or the wider phenomenon needs kind of something else. And as I mentioned just a few minutes ago, you also have pretty patchy settlement evidence from this kind of middle Neolithic, mid 4th millennium horizon onwards. So this is a lovely example. It's not from Ireland. It looks like it might be, because it's really, really similar settlement evidence. This is the site of Clonfeithley on Anglesey in northwest Wales. So you see here in red the really typical early Neolithic kind of timber house footprint, that rectilinear thing, very, very classic. But overlying that and marked out in yellow, is what you get an awful lot of across Ireland. And in this region too, you've got this very kind of confusing arrangement of pits and spreads of post holes and scatters. So very more difficult to interpret archaeologically, but still present. So it's something that needs to be linked up. And again, as I'm sure many of you know, excavating prehistoric sites in particular, bone preservation is such a huge problem in acidic soils that you find across Ireland. So you're dealing with that decay, that differential preservation that you're not necessarily getting in other regions. So that's something I think which hinders wider analysis or earlier analysis before then. So what this project is doing is to kind of dive down or dig down into the molecular level of information. So the kind of the guiding principle of this project is that lots of really good and interesting and exciting information is still preserved. It can still be preserved at the molecular level. So it's stuff that's not always visible macroscopically or identifiable in the kind of traditional techniques. But if you analyze these with scientific instruments that look at kind of basically individual molecules, you can extract that information that is still kind of hunkering in there and surviving, even though the wider object or the pottery sherd looks quite kind of battered and kind of destroyed. So we're looking at basically three kind of categories of material. We're looking at pottery sherds, so ubiquitous, fairly robust, they hang around for a long time, even though they might be kind of broken up. Bone fragments, and these are specifically kind of fragments that have either been so degraded or broken up or kind of semi-dissolved that a traditional zoo archaeologist couldn't identify, you know, what species it is, because you don't have the size, you don't have the intact bone, I suppose. So previously, a lot of those bone fragments could only be just basically described as burnt or unburnt bone fragments and just kind of bagged up and left in the archives that way. So we're tapping into, again, the information in the collagen that's still in that bone to potentially identify species. And I'll show you how we're doing that in a second. Another big category of information is teeth and bone, but in particular teeth, because tooth enamel is much more hard-wearing than bone, and that can survive even when other bits of the skeleton can't. And there's a lot of chemical information potentially that's locked into that tooth enamel as it has been laid down, either when the child was growing or the young animal was growing. So that you can go back to that and tap into it. So again, I'll talk about these techniques in a bit more detail. So the first one up is lipid analysis, and Donald mentioned this in his intro. I had a fair idea going into this project, well, a very good idea going into this project, that lipid analysis was really, really useful for Irish material in particular, because we don't have that survival of animal bone that you can kind of reconstruct husbandry practice and things like that, and knowing that a lot of information you can actually get from the pottery sherds. So I had looked at a range of Neolithic vessels from the early to the late. I think on that map here, the early is in red, middle is in blue, and the late Neolithic, the grooved ware, is in black. 13 sites, I think, a while ago now, nearly a decade ago now. Anyway, a range of these vessels, and nearly 500 of them across a range of site types. And the fabulous, we didn't realize going into this in 2011 that the preservation was magnificent. It looks like, kind of conversely, the acidic soils, which are so problematic for bone, for preserving bone, actually enable the better preservation of lipids in the pot. So there's the soils, and I think the Irish climate as well, you don't get extremes, so it's not very hot, it's not very cold, quite temperate, quite damp. That's actually quite good for lipid preservation. So 90% of the sherds I looked at in that study had residues, archaeological residues, surviving. So it was an amazing kind of repository. And again, just tackling stuff, small pieces, 2 to 3 grams is all we kind of subsample from a sherd, grind it down to better extract the lipids in there. So using what we call kind of a dry cleaning technique, where basically you're just using solvents to extract those lipids, kind of make them jump into the solvents and leave the powder, and then you analyze that solvent mixture. And I don't know, I still find these chromatograms interesting. They mightn't make a lot of sense to people here, but what we're showing here is basically a lipid mixture that's just been separated out, so you can see the individual lipid compounds, and basically those collection of lipids are telling you that that is a classic sign of a degraded animal fat. So really high in free fatty acids, and lowish in these triacyl, diacyl, and monoacyl glycerols, which are kind of the parent fats that would be there in fresh fats that have just, degraded over time, and you get loads of these guys. But anyway, if you saw it in the lab, you would immediately go animal fat, degraded animal fat. So lots, 60% of those residues that I got in that earlier study were degraded animal fats. So there was brilliant preservation, very exciting. And that's just a little test tube, or not even test tube, a little vial, a 7 ml vial. So it's about, 3 centimeters, 4 centimeters high. These traces are so tiny that you can't normally see them with the naked eye. They're invisible, which makes for a very weird sensation when you're analysing things in the lab, working on things you can't see. But every now and again, the lipid was so concentrated that it did actually start to become visible. And this is just one of those little vials there where you can hopefully see little rings of lipids kind of clinging to the vessel wall, or to the vial walls, which kind of tells you that's a very rich sample. Anyway, how do you analyze them more, or how do you tell kind of what sort of degraded animal fat you have? Well, happily, because I suppose because the wider carbon cycle and how carbon works its way around our bodies as organisms, as a human, or as a kind of a herbivore, or a non-ruminant herbivore like horses, basically the different carbon isotope values will fractionate differently. They will take it up differently. And basically that results in a situation where you can tell you can distinguish ruminant, so cow, sheep, goat, dairy fats from ruminant adipose fats, so their carcass, their body fats, and you can kind of further separate out equines and pigs, for example, monogastric animals plot in a completely different area. And that's basically due to how carbon will circulate through an animal that has kind of 4 stomachs, and it'll all kind of do its different things versus kind of single stomachs. So it's really interesting. It's something that's been, I think, that was noted on in the veterinary world, or reported on for a long time before it was kind of applied into lipid analysis. But that's how we tell different fat types apart. We look at their carbon isotope values, we measure those. And the significance, I suppose, for somewhere like Ireland and Britain is that looking at huge collections of these Neolithic pottery sherds, we see there's a really strong dairy signal. So you see here, green is dairy, and pink is carcass or adipose fats. And you can just see here, just even eyeballing it, there's a huge percentage, a huge proportion that have a very strong dairy signal. So dairying is definitely well embedded in the Neolithic, from the Neolithic onwards in Ireland. And that's what I was looking to kind of run with or explore in more detail in the Passage Between People project. So did that signal kind of continue on basically through the Neolithic, and maybe is dairying one of these subsistence strategies that's underpinning passage tombs, for example. So it's not so much about the cereal agriculture, the arable agriculture. It might actually be kind of more livestock based. So this is one of the hypotheses. Another nice little side one, which we're kind of digging into a bit more in the Passage Between People project, is to look at what's in the ritual pots. So it's something I avoided in my earlier study because we weren't sure we'd find anything, and it was a bit of a risk. But now we know that the lipid preservation is so good in Irish pottery, it's definitely well worth examining vessels from quote unquote kind of ritual context. So you have some examples here of the very famous Carrowkeel bowls that you find in passage tombs. And there were just two that I looked at in that previous project, which came from the Ballynahattie complex up in Northern Ireland. 2 Carrowkeel bowls you see here from kind of kist environments and kind of subterranean kists. And one of those, one didn't have lipids, but one did. One contained degraded animal fats. So definitely you can find stuff in these kind of ritual bowls. They're not necessarily just for containing things, or putting kind of cremated bone into a deposit. They can frequently have been used prior to their deposition, which is a nice discovery. So the second strand looks at these bone fragments and proteins. And again, just to give you a bit of background, general background into kind of bone structure. Bone is made up of minerals, water, and proteins. Okay, so most of it is minerals, the mineral structure, but a smaller percent, about 30% or so, is made up of proteins. And of that, most of these proteins are collagen, okay? And collagen holds the information, or holds a lot of information on species and types. So if you can break down bone fragments, and you don't need a complete bone, you don't need an intact bone, this is ideal. This is something myself actually and Michael Buckley sampled from Nouth a few years ago. We had these unburnt little fragments which had been labelled or tagged as animal bone, and you can extract the collagen from these small little fragments and find out what species it does. So it's basically just a procedure of dissolving the bone in acid, separating out the collagen, and in a way that you would do with those lipid mixtures I showed you earlier in the pottery sherds, you separate out the collagen in that same way into the peptides, and from the patterns in those peptides, basically you can fingerprint the species, so you can tell what animal you have. These are just some of those fingerprints. Basically, those different peak intensities are giving you information on the species. So there's a whole range. Initially, the range of animals started out as quite small, because you obviously need reference animals, so you can match up your archaeological signature from your reference signature. So it was initially very useful at telling sheep-goat apart. So any zoo archaeologist will tell you the similarities of sheep and goat bone. It can be sometimes quite difficult to visually separate them out. But at this molecular level, at the level of collagen peptides, they kind of differentiate themselves quite nicely. More recently, it's been widened out to kind of wider ungulates, so kind of deer and things like that. We can tell humans apart from animals and marine mammals. So that range of animals is kind of ever expanding, basically, as people do more and more work. And this is all kind of the work of labs like Manchester and Michael Buckley and York. They've done analysis on some very, very old bones, so 100,000 years plus, and they've got secure IDs on that, so it can be very old. And they've also tested stuff on quite degraded, quite heat-treated bone, not burnt. You can't do it on burnt bone, but you can do it on bone that's been fairly bashed about a bit. So it's a really nice type of analysis. And as I mentioned, this was a little pilot study we did back in 2015, got a bit of funding for the Royal Irish Academy to do it. And we targeted some secure kind of contexts of kind of unknown bone fragments in Nath, the Eastern Tomb in Nath. And you can see here, this is kind of the report of the zoo archaeologists, so that the morphological stuff. And you can see here, I've highlighted them in red. Nearly all of those fragments were unidentifiable macroscopically. So, you couldn't tell if it came from a cow or a sheep or a human or whatever. So just a few small IDs there. So a lot of them. And we targeted, with the help of Kerry actually, some secure Neolithic contexts that we knew we were going to go after the Neolithic bone. 150 odd samples in the end from both the right and left hand recess, this Eastern Tomb. And the lovely thing was, I'll just flash up some of these. We got some really nice IDs. You see here, human was mixed in there. So some of those small bone fragments were actually human. Some hare, cow. You see the success rates there, fairly high, 53 out of 88 IDs kind of didn't fail. So so I suppose the take home from that is, that had been previously been labeled up as animal bone, because understandably the zoo archaeologist couldn't ID that to a species, but we found an awful lot of that was actually human bone that had been processed and deposited in these kind of pits and things, these cuts in Easter tombs. So very interesting insights into kind of mortuary practice from using zooms. The final strand, just keep an eye on time, we're good, yeah. The final strand is looking at all the kind of information you can potentially get from bone and teeth, from tooth enamel. I'm sure loads of you will be kind of aware of the marine terrestrial kind of differentiation you can make if you look at bone collagen. You can tell if a person, either a human or an animal, has been kind of chomping on, say, marine resources or terrestrial resources by looking at particularly the nitrogen and carbon values in their bone. So you can see here, this is a plot of carbon and nitrogen, and just so you can kind of pick apart that plot there. Humans are the open circles. Sheep are the triangles, closed triangles, and this is generally the patterns you'll get because humans are kind of omnivorous and they're kind of eating things further down the food chain. They tend to have the most elevated nitrogen values. But with the carbon, what you can tell is the less depleted, so minus 16 is less depleted than minus 23, you can actually tell how much of that food source is coming from marine versus terrestrial. So less depleted carbon values, so higher, will be more fishy, more marine-y, more aquatic, and more depleted will be more terrestrial. So you can see here these humans are actually sitting in quite a terrestrial place, whereas these are sheep values that have quite marine values, and they seem to be eating resources like seaweed. So you can start to pick apart not just human diet, but the diet of animals as well. So were people doing things like foddering using marine resources like seaweed? And that is something that we know is very well established in places like Orkney. So we're exploring those sorts of practices in Ireland as well, the extent of those practices. Oops, sorry. skipped forward there. Well, it's not recent now. I suppose it's about 10 years old or so. This technique was pioneered by Marie Bellas, the collaborator I mentioned at the start, working in Paris, where she has actually done sequential analysis. So instead of getting a bulk reading and just saying broadly what an organism was doing over its lifetime, if you take incremental samples along a growth axis and you measure it and model it, you can actually get growth curves or seasonal curves, and you can see values potentially go up and down with the seasons. So again, I appreciate these plots are a bit of a mind boggle if you're not used to looking at them, but if you can see here, the black dots are carbon values, and the clear or the white dots are oxygen, and they're all kind of sequenced. And you see here that the carbon stays pretty much level through the growth axis, which is kind of the time. It's over time, whereas the oxygen goes up and down, and that is showing you the oxygen is a record of water that you take in during the year, and the oxygen values on that environmental water, vary with seasons. So it's basically a proxy for the different seasons for summer, winter. But you can see here that carbon stays pretty much flat, which is telling you that through that seasonal cycle where the water goes, it's different values, that animal has been eating more or less the same things because it's got the same carbon values. But you see here on this lower graph, in the seasonal oxygen loop, those values go from being quite, relatively high to relatively low, which tells you there's been a switch in food sources. So that animal at that point of the year is eating something quite different from what it's eating at that point of the year. So again, it gives you an indication of seasonal changes, which is really exciting for animal husbandry in the past. And the very final thing is just strontium isotope. And again, I'm sure quite a few of you would be familiar around what it can do about kind of movement or informing us on movement. Strontium, again, is one of these chemical markers that is locked into the tooth enamel, the developing tooth enamel in the early years. So whether you are you're a calf, or you're a piglet, or you're a child, and when the tooth enamel is forming, where you are, on what geology you are while you're kind of eating and drinking, those chemical values are being absorbed into your tooth enamel. And when your tooth enamel stops forming at a certain age, those values are locked in. So if you then move in your lifetime, whether you're a cow, traveling to your final resting place or to a market, or you're a human traveling somewhere else, if you have moved onto a different geology, different area, there'll be a difference in those values. So you can kind of measure movement in that way. So again, in the Passage to People project, we're mostly focusing on the movement of animals because we want to see or want to investigate if there's a link between these passage tombs, which seem to be kind of referencing a wider environment, a wider territory, they're built up, in high ground. If there's a link between that and kind of the wider movements of people with their animals. So, are they exploring wider territories? Are the farming practices kind of wider in the landscape? So that's how we're using that there. In total, again, just to sum up, this is the kind of stuff we're looking at, the kind of volumes, I suppose. And the nice thing about these different approaches is you can kind of interconnect them. So in the 3 different zones we're looking at, in Northwest Wales, in Ireland, in Orkney, the level of preservation is different in each area. So Anglesey, for example, massive kind of granitic bedrock, so very, very poor bone preservation, even kind of worse than Ireland. So we're not going to get a lot of bone there, but in that zone we focus more on the pottery to give us information on kind of the animal husbandry practices and things like that. So you can kind of use these techniques to give you coverage in areas where you don't have coverage via other means. So we're looking at, large number of animals and humans using kind of isotopes, carbon, nitrogen, kind of oxygen diet and things, strontium for the movement, the lipid analysis I've talked about, and giving kind of species IDs. And we're also using, obviously, because not a lot of these remains come with very secure dating as it stands, we've got kind of a budget for 150 radiocarbon dates. So just to finish up with, excuse me, I'm going to give you two small examples of kind of ongoing work packages within the project. So one is looking at the pottery assemblages that we're uncovering in North Wales, that's being done in Bristol, and some really exciting work done on a site actually excavated along the N2 motorway, or the M2 motorway, a few years ago now, I think 2004 or so, in Dublin. So that's kind of a direct parallel for maybe some of the material you might be uncovering on the N55. So first of all, this is the work of our PhD student Lily in Bristol, supervised by Richard Evershed. So Lily is looking at a lot of pottery assemblages from Anglesey, in particular the island of Anglesey. Anglesey has seen a lot of, well, it's gone from an area where there was no excavation, or there was kind of antiquarian investigation into the megalithic monuments, but no kind of open plan stuff, to a period over the past 10 years where there's been quite a lot of development. So roadways, motorways, a couple of big industrial parks kind of sites, so big kind of open plan excavations where they're getting the sorts of material that we've been getting in Ireland for quite a while, kind of infrastructural stuff. So she's got quite a few assemblages that are uncovering this, these lovely pottery links that kind of connect to the stuff we find in Ireland, in Eastern Ireland, but are also quite different. So this is a lovely vessel type called kind of Fengate ware, you know, that flat bottom and that kind of tapered base, which is something we don't see in Ireland in the Neolithic. It's very common in Britain. So Wales seems to be a bit of a zone at the moment of kind of a crossroads or interaction where you get settlement evidence which looks like really similar to Ireland, but at the same time you're getting these elements of a material culture that aren't found in Ireland. So it's quite a nice mix. So Lily is looking at the contents of this pottery. As I mentioned before, Anglesey is really bad for bone preservation, very, very poor, very patchy. So this is one of our main ways into assessing what these communities were doing, how they were surviving, what kind of animal husbandry practices they were undertaking in the Neolithic at the same time, you know, as we have these passage tomb sites, excuse me, over in Ireland. And you can see here, this is just some of her initial results. This, as far as I remember, it's been a while since I looked at these. Yellow are the grooved ware pottery sites, so quite a strong dairy signal from those. And this is a mix of the Fengate ware and this, what they call Mortlake ware in Britain, but it's kind of our Middle Neolithic impressed ware bowls, so very, very similar types. So nice patterns there. Also, and she's having a good bit of success with dating. So the lipid extracts that are taken from the pottery, actually directly dating those lipids, which again is another immensely powerful way of, I suppose, accessing dating material when you don't have other traditional sources like bone, like charred plant remains, that sort of stuff. So she's actually, or she will be doing it at the moment, be able to directly date these pot types, which will tell us a lot more about the exchange of traditions and influences across this Irish Sea zone. So it's really exciting. And just my final slide is the current work that we're undertaking on a site that was actually excavated, as I said, on a motorway scheme, on a TAI scheme, which had fabulous zoo archaeology done in it, you know, as part of the post-excavation program. It's actually a site that you can potentially extract even more from. So we've gone back to the assemblage. We're particularly interested in it because Ireland has such poor preservation of bone in general. This is one of the big sites that a large collection of animal bone has been recovered, and nearly 60 cattle are found to have been deposited in this ditch. Again, the dating is kind of patchy because the collagen quite poor in this assemblage. They couldn't, when they tried to date the bone, they didn't get a hit rate on the bone. So we're trying other strategies to date that now. But if you look at where Kilshane is in North County Dublin, it's about 20-25 kilometres from the Boyne Valley and places like Newgrange and Nowth. So it's in an interesting position. Our zoo archaeologist Fabienne has added to the bone analysis already done. So she's done some initial additional calculations on molar length, so tooth length. She's looked at the phalanges, so the foot bones, looked at wear and pathology, and is actually coming up with some really nice evidence for bone wear, which might be indicative of traction. So some exciting results that these animals could have been used for pulling large weights now, whether that was ploughing fields or pulling big stones to build passage tombs, we're not sure yet, but it's really nice that additional analysis, you can go back into it. And we're also going to be undertaking strontium isotope analysis on these animals, again, to get a sense of how far they're moving, who or what or where are the communities from that are bringing this large number of animals to be slaughtered and deposited in these ditches. Are we talking about a big kind of widespread community, or are they quite local? And we're also doing lots of this sequential isotope analysis on the teeth to get an idea of seasonal resources. So are they being fed different things at different times of the year? Is there a birth season with that? So very, very exciting bundle there. So yeah, just to conclude, this is, hopefully I've kind of illustrated how using lots of excavated material from multiple assemblages, so they're not all passage tomb sites, they are sites that are contemporary with it, and you can use all of these to build up to a bigger picture of kind of what's going on in the time period. So that the hope for the project over the next year and a half or so is that we get this better understanding of these kind of hidden communities currently behind passage tomb construction, and get a much more sophisticated idea of farming along this Atlantic fringe and how sophisticated people were. And crucially, I think, is to that really nice kind of, I suppose, side result of this is that all these techniques are kind of, you can add them together to kind of generate a nice toolkit that can be used to target particularly challenging horizons or archaeological remains. So I'll just end it there. Oh, I think you're muted, Donald.
Thanks very much, Jessica, for that. I'm sure everyone will agree there was an awful lot of information in there that's going to take time to digest. But I suppose one of the things that came across very strong for me there was up until now, we would have been quite dismissive, I suppose, of some of our non-cremated fragmented bone, because our zoo archaeologists could very rarely do anything much with it, and wouldn't end up telling you an awful lot. And it's great to have some options there for further study in the future. I have a newfound respect for it. I might just go over to Kerry now, Jessica, if that's okay, if you have some time just for some questions. Maybe just open up the questions, and then if we've time, we'll go to the floor. Kerry, can you come in there?
Perfect. Thanks, Donald. That was brilliant, Jessica. Thanks a million for all the information. Before I move on to questions, I'll just give people a couple of minutes to add some things to the Q&A section. I'm just going to share a slide here as well that Dr. Julie Dunn sent us on for this seminar as well, just in relation to the actual getting the analysis done from a commercial archaeology point of view. So they're still obviously early enough days, but they're putting together some flyers at the moment. So she's hoping to distribute those when they're done. But this just gives you a sense of, say, roughly how much it's going to cost per sample. And they do both the residue analysis on the pottery, but also on the burnt flint or stone, which obviously is something that Jessica couldn't go into herself, but it's a very sort of early days in the process of looking at the stone samples for the lipids as well. So it's interesting that some Irish samples have been going forward for that as well, to get that underway, including some of the samples we've done ourselves. And then I know other commercial companies have also been sending samples to Julie. So it'll be interesting to see how that develops in terms of burnt mound sites and how things are sampled on site for that. So it'll be very important from an Irish point of view to see how that develops. But that's just to give you the idea for her contact details there and so on. Basically, if anyone has any particular queries, Julie said if you email her, she'll arrange a call and discuss with you basically the best process in terms of sampling for your particular site and how much material she needs for the analysis and so on. So it's really a great service. Okay, we'll probably open it up to some questions. So hopefully people can add to the Q&A box at the moment. We'll just start off with one here, Jessica, if that's okay. So it's back to using the zooms analysis, and it's just about whether or not there have been any particular species identified as being frequently misidentified, perhaps.
Yeah, good question. Not so far. The IDs that we've got back so far are kind of confirming your classic kind of triumvirate. So cattle, sheep, goats, excuse me, in there. You see, so I think at the moment it's kind of especially useful in scenarios where you have no prior information, if you know what I mean. So you just have kind of a collection of fragments and you're like, it might be cattle, it could be sheep, not sure. So it's good for seeing the presence, absence, the range of species you have. Though interestingly, if you have, particular contexts and you thought, I wonder, is that, a human burial or a human deposit or is an animal deposit, it can be really nice in picking apart that. The NOWS pilot study was quite a nice surprise. We were just expecting to get a range of animals, because normally, or the received wisdom is that, animals don't get burnt and humans get burnt in these sorts of scenarios. So it's nice to see that being kind of turned on its head a bit. But at the moment, mind you, it's still early days with our zooms analysis. We haven't undertaken a huge range yet, but the hits we're getting are kind of confirming, I suppose, what a morphological approach would take, like as the traditional zoo archaeologist.
Okay, and following on from that, can the bone that is slightly charred be also used, or does it have to be completely unaffected by fire?
It's not looking good for that. Again, with the NOWS pilot study, we sampled a range, excuse me, of bone, that looked visually affected from, your kind of creamy, unburnt, through to kind of blackened, through to kind of calcined, and really anything that had showed signs of burning didn't have a good success rate. So I wouldn't hold out too much hope. So that said, the cost of that analysis is relatively cheap. So it's in around, I think, 15 or 20 pounds, something like that. So it's not like a radiocarbon date, for example, where it's 300 quid or so, and that's a lot of money if you're getting multiple samples. So I think it's affordable enough that you could take a few gambles with material you weren't too sure on. But generally speaking, the charring, that sort of heat destroys the collagen, basically.
Okay. And sorry, maybe you might not know the answer to this, but do you know if both Manchester and York kind of do it commercially, or is it primarily built into research projects?
Yeah, I'm not too sure about the situation in York, actually. I know they have done it in the past. Our arrangement with Manchester is a research arrangement. But that said, Manchester are involved with a lot of projects, and I see the papers come out, like there are lots of different things. So I think they're well used to working with targeted sites or targeted assemblages, or not smaller questions, but you know what I mean? It doesn't have to be a big kind of multi-million and whatever project. They can answer some really interesting questions about resource use. A lot of lovely, interesting work in recent years about marine mammals and things. So going back to ambiguous assemblages and identifying kind of whale and seal and all sorts of things, or even artifacts as well, where you're not sure. was it bone or antler or what it's made out of? Because again, so many of these techniques sample such small amounts. You're talking about milligrams, that it's quite a nice option for exploring materiality and what things are made of.
Mm-hmm.
So it's fairly nice.
Yeah, I suppose on that note, we should obviously emphasize that everything is done under license.
Oh yeah, absolutely. And unfortunately, post-Brexit, there's an extra step now. Mm-hmm. So any of our analyses that will be undertaken in the UK that we haven't already exported, we will need to get additional non-EU permissions. So the advice from the museum is that it's taking an additional number of weeks to get approved because it's a separate department. So I'm sure people are aware of that already if they've been encountering
the paperwork is piling up indeed. Okay, we've a question in there from Ken, which is sort of linked to, I suppose, the previous one in some respects. So it's about the dating of the lipids, and that question about whether or not so far, maybe this hasn't, I know it's quite a new one, so have any of the dates returned so far conflicted with any C14 dates?
They've supported it, I think. The nice thing about the lipid dates is that there's a really strict quality control thing of them. So that's good, obviously, but it also means that they can discard a lot of dates and samples because they're not entirely sure. So what you do with the lipid date, you extract your lipid, and those two fatty acids I talked about, the C16 and C18, they are both independently dated, and they need a sample where those proportions are the same. You've got roughly the same amounts of C14 or C16 and C18 fatty acid, and because if they are in differently, oh gosh, I'm telling myself I'm not there. If they are in massive different proportions, there's a potential that the radiocarbon date on one is not going to be the same as the other. So basically they have to be the same proportions, same ratios. You get two dates and those dates have to agree. So if you've got a radiocarbon date on the C16 fatty acid that's different to the other one, that date is discarded. So it's like, it's a bit sketchy, we're not going to use it. So you need agreement across those two fatty acids, and then it works amazingly. It can provide you with a date where you've no other dates, which is great. It can tighten up existing chronologies because if you think of what you might normally pull from a site to date, a bit of charcoal, some plant remains, a bit of bone, sometimes that association or relationship with the event you want to date is kind of weak. It might be a bit of charcoal in a pit and you don't really know how the charcoal got in there in the first place. Whereas if you're dating lipid in a pot, the pot is archaeological, was from a certain time period. So dating that lipid in the pot is a really strong kind of association. So when it works amazingly well. Like I think when they were testing it, and that's in the recent Nature paper that kind of publicized the method, they went back to the sweet track in the Somerset Levels, which is Neolithic trackway that has two or a couple of carnated bowls chucked over the side, and obviously the bowls are Neolithic identified, and there's a dendro date on the sweet track, which is 3807 or something super precise, basically 1 calendar year. And when they applied the lipid dating to those pots, they got within 5 years of that dendro date. So when it works, it can be amazingly precise, because the probability is reduced, because it's from that pot itself. That said, though, the caveat is there's a number of steps. So first of all, you might have a sherd that you want to date. You've no guarantee that there's enough lipid in that sherd to date. And okay, if you can extract the lipid, the fatty acids mightn't be in the right proportion for that date to proceed. So there's a couple of kind of hurdles that a lipid date would need to get over. Yes, it's not guaranteed, but for example, we're working on some pots from the Cage of Fields at the moment, stuff that's just come up out of excavation past number of years. And I think we managed to get 4 or 5 dates from, I think, maybe 35 samples. So they weren't all tried, they weren't all dated, but out of the lipid analysis we did on 35 sherds, 5 have turned out to be suitable for dating. So that might give you a bit of it's probably, 1 in 5, 1 in 6 chance of being a suitable candidate for rotary carbon dating. But again, Julie is in Bristol, is a really good contact point for that. They can give you a handle on feasibility, I suppose, of that approach. But it's really exciting.
Yeah, it's obviously very new, and those are quite high odds, actually, probably higher than I would have expected them to be. Another question from Ronan, who's said, great presentation, Jess. Basically just wondering what your sort of key recommendations would be for excavators that are currently excavating sites. Like, what are the sort of pitfalls maybe that they should avoid in terms of their sampling, or perhaps when they're looking at these types of methodologies for research questions?
Yeah, I think the biggest one, to be honest, of the material that we're working with now with passage stream people, some of it has been recently excavated, but a lot hasn't, and a lot of it's kind of decades, maybe generations old, and obviously had been taken out of the ground not knowing what could be done about it in the future. So I wouldn't say that sampling or poor sampling or not, whatever is detrimental to this analysis. I would actually say the important thing is research questions. So making sure that you have a reason for that type of scientific technique. There's no point in, say, getting in a zoo archaeologist if all your bone is kind of scrappy and fragmented. It might be better to use zooms for that, for example, protein analysis or lipid analysis, if you've no bone at all, because you want to get an angle on that. Also, sample size does matter. So data sets, so kind of there's basic kind of stats and statistical minimums for confidently assigning something. So Bristol, for example, will always say, give us 30 sherds, because you kind of need 30 data points to confidently describe, or you need 30 samples. So things like that. I think that would be the biggest pitfall, kind of undersampling, only submitting, 1 pot or 2 pot sherds for lipid analysis. And okay, you might get something in them, but what does that tell you about the wider site? So it's scaling your analysis and your sample appropriately to answer the question. It's kind of that. I would say pretty much like those materials, they've obviously been in the ground for a very long time. And with pot sherds, okay, you can get maybe a bit of contamination, a bit of transfer from, say, finger lipids onto a surface of pot. But generally speaking, all these surfaces are cleaned anyway before analysis. And it would have to be something pretty drastic to kind of interfere. Okay. But beyond kind of normal procedure of maybe wrapping something in foil before you kind of bag it in a plastic bag, that sort of thing. But generally speaking, and in terms of kind of teeth and bone, as much as possible, it depends on the nature of your bone. Is it loose? Is it ex situ, in situ? Is it connected to a skeleton? Like recording, obviously, common sense detail like that, like where in the mandible it comes from, or is it connected, things like that. So I'd say it's more about choosing your research questions and choosing the number of samples you want to get analysed would be the biggest, would have the biggest impact.
Yeah, and I think it's probably worth reiterating again, Julie, as I said, is very much open to answering these questions too. So, and like certainly when it comes to the burnt mound material, which as I said is quite new, she has specific recommendations for people in terms of how they sample those as well, in terms of if you're looking at mound material, bottom, middle, top, and particular size of stones, and things like that, and then taking a decent enough sample that then they can select what they think is the best in the lab as well. So I think it's probably, there's a lot of advice out there. I suppose that's the point.
And they're happy to field those questions. Like, it's only a quick email usually to just check before anything comes out of the ground. In terms of the lipid analysis, and I think you showed it in that previous slide there, Bristol have, in partnership with Historic England, have come up with a really great guide for residue analysis, organic residue analysis, which answers all those questions about how should I sample, what sort of questions can it answer, when is it useful, when is it not. And that's a PDF and open access. That can be easy circulation. It's a good protocol.
So just another one in there from Ed. He just said again, what was roughly the cost of the lipid dating? So given that they will potentially have to look at 5 or 6 samples to get one date. So I think we were estimating, well, Julie was estimating around €100 per sample.
Oh, for the lipid analysis, yeah, the date is, so there's a couple of steps at that.
Yeah.
What probably would normally happen, or ideally happen, you'd have a collection of pottery sherds that you'd get your lipid analysis done on. So, you know, what's in them, are there lipids, what sort of lipids? And that's the kind of 100, 150 mark. Out of those, there might be a few samples that are really, really lipid rich and worth dating or have potential for dating. So in that scenario, because you need more lipid to extract more lipid, because basically like carbon-14 is like a tiny amount. You need to extract much more lipid to get the carbon-14 you need to measure. So they do a second extraction on the potential, you know, the sherds with high potential, and then date the C14 fatty acid or C16 fatty acid and C18. So it's effectively 2 radiocarbon measurements for the one date. So it's twice the price of your standard radiocarbon date. So, you know, it's not cheap. Yeah, you're talking about 500 to 600 pounds probably. So I wouldn't say it'd be your first port of call. You know, if you have more suitable, if you have other material like bone, chard plant in a nice association, a nice context, it's what's cheaper. But if you either really, really want to know the direct date of your ceramics, or the ceramics is kind of the only material left to you to radiocarbon date, then I'd probably go down that route, kind of you know, after the first route, you know what I mean? So yeah, it's worth bearing in mind. It's very achievable, it's very doable, but it is expensive. So it's probably not your first port of call if you want to work out a chronology for your site. Yeah, but again, Julie can give you precise updates, but it is around about that. Yeah, up to kind of £600 for the combined kind of date, pottery date.
Yeah. Okay, great. Okay, just might see if anyone has any other question. I might just ask one as well while we're there. You know, we're talking about meaningful sort of sample sizes, and just in terms of actually, say, the isotope analysis. You know, in terms of, obviously we're looking at it from a commercial point of view, and so, you know, are a couple of samples from one site really going to tell us much, or, you know, do we need to start looking at these in broader pictures, like, you know, is data from other sites available? How do we compare? That sort of question.
Yeah, I'd always recommend doing that first, doing a literature search of what's been found out, because it's one, especially with the incremental sampling, again, because it's multiple measurements, even on, say, a single tooth, that stacks up price wise, even though you know, the cost of a carbon nitrogen run could be kind of maybe €15, €20 if you're doing that 10 times across one tooth, and then say you might have 5 animals, 10 animals, you know, that's getting into the thousands. So it always, check on what's out there, if you feel strongly that your samples will add to that picture and what they might add, the potential to add. Generally speaking, when those animal studies have been undertaken, you're probably talking between 5 and 10 individuals they look at, because less than that, fewer than that, you don't know how representative it is of the herd or of the site or of that community of animals. So it's a bit like a random hit. So you do need to be fairly confident that you're kind of describing the wider population, and that's why, again, sample sizes need to be above 5, I would say. But again, looking at kind of recent isotope papers, you get a handle on what's been the sorts of numbers people typically think is of publication standard. Sometimes you do get people kind of drip feeding their analysis, so they'll do a whole site and they'll kind of publish the humans and then they'll publish the animals and they'll do something else. But you can get a very quick sense of what's an acceptable data set, I suppose, for analysing. And it's, I'd say probably in around 10, between kind of 8 and 10.
Okay. Okay, I think maybe last chance if anyone has any particular questions or want to raise their hand to come in. I think everyone's you must have answered everything, Jessica. Okay, I might hand back to Donald so to finish up.
Jessica, could I just quickly ask? I know I'm conscious of the time here, and I know this isn't your area specifically, but other than ceramics, do you think is there options maybe in the future for stone? I know wood is really out, isn't it, in terms of lipid analysis? Do you think is there any potential for stone to absorb some lipids that could be?
Yeah, I think as Kerry mentioned, it is starting to be explored a bit in the philosophia kind of context. Probably needs a fair bit more experimental work. So all that lipid analysis on pots, it seems so kind of straightforward and understood now, but that's, you're probably talking about 30 years of PhDs and experiments and all sorts of things. So it takes a certain level of experimentation on a new material or a new kind of category of object before you can be really confident that it works. So the potential is there. There hasn't been just enough detailed work, and for that you really do need things like a PhD that can kind of mine down into it over 4 years and cover lots of angles before they can kind of come up with things. So I'd say it's there, but needs more work. Because there's different variables to examine. The absorbency, how it clings, how vulnerable it is to contamination, what's the influence from the burial environment, all that sort of stuff. It's kind of been gradually being worked out for ceramics and is only just starting for things like stone.
Just to reiterate that, it's essentially what Julie has said is that it's early days, but that they have had some success, all right, with some Bronze Age Fullochthea sites in the UK, but maybe less success with some of the Irish ones so far. So it's about interpreting, I think, so far at the moment. She's looking at how we're interpreting maybe the lack of lipids in some of our burnt mound sites, and obviously they're complex multi-purpose sites, and in terms of if there was boiling of meat and so on, when within the life cycle of the site that took place. So I think it's early days, but certainly something to feed into.
Okay, well look, thank you very much, Jessica. If there's nothing from anybody else, I'll bring it to a close because I'm conscious of the time. And Jessica, thank you very much for taking the time out. I know this is a very particularly busy time in the college, so we appreciate that this morning. Thank you very much.
Thanks very much, and thanks for the invite.
And thanks very much to my panelists, to Niall, project archaeologist for the scheme, and to Kerry for organising the whole thing. So I hope you all enjoyed that. Thank you all for attending, and we'll see you again soon. Thank you.
Bye-bye.